Hello, and welcome back to the Deep Dive. I'm Adam.
And I'm Grace.
And yeah, we're your grade 8 science teachers here at School on X.
We are indeed. And today, we are so excited to kick off a really, really fascinating deep dive
Totally fascinating.
into, well, a core topic for you guys in grade 8 science. Chapter 6, Reproduction in Animals.
Yeah, it's one of those absolutely fundamental processes in all of biology, how life, well, how it keeps going.
It's incredible when you stop and think about it, isn't it?
Mm.
I mean, just imagine for a second, what if the world just stopped?
Stopped having babies.
Yeah. No new generations. It's like a chilling thought, right?
Definitely.
So what's the process that keeps everything going, keeps the tapestry of life vibrant and unbroken, species after species?
Gotta be reproduction.
Exactly. And our mission today really is to unpack the incredible ways animals make sure their species continue. We'll be sort of pulling apart the pages of your science sources,
Getting into detail.
giving you those essential nuggets of knowledge, those aha moments.
Hopefully a few of those.
And maybe even a few surprising facts that'll make you look at the animal kingdom with, like, totally fresh eyes.
That's it, exactly. Because, reproduction isn't just another biological process on the list. We talk about digestion, circulation, respiration,
Uh-huh. All vital.
all absolutely essential for the survival of every individual organism. You need them to stay alive right now.
Right.
But reproduction, that's different. It's not strictly about your individual survival.
No, it's bigger than that.
It's about the survival of the species, the whole group. It ensures we get continuation of similar kinds of individuals, generation after generation. It just keeps life renewing itself.
Precisely. It underpins, well, pretty much all of biology when you get down to it. It shapes ecosystems, it drives evolution, it creates all that stunning biodiversity we see out there.
It's huge.
And today, we're going to get into all the crucial details of how this amazing process happens, specifically in animals, exploring the incredible diversity, the ingenious strategies life has evolved.
Okay, so where do we start? Let's begin our exploration by looking at the two main paths, the two primary ways to make new life.
That was good.
Just like we saw when we looked at plants, animals have basically developed two main strategies to create offspring. Two really clever ways to ensure their species not only survive, but, flourish over time.
Okay, so what are these two fundamental approaches?
Well, these two fundamental strategies are sexual reproduction and asexual reproduction.
Right, two big categories.
And while both get the job done passing on life, they use vastly different mechanisms. You can think of them as like two distinct evolutionary pathways.
Each with its own set of pros and cons, really, depending on the animal and where it lives.
Exactly. So let's start by properly defining sexual reproduction. What's at its core?
Okay, so sexual reproduction, at its very core, this mode is all about the fusion of specialized cells.
Specialized cells.
We call these unique cells gametes. And the key thing here is that it inherently requires the involvement of two distinct parents, usually a male and a female.
So it's a team effort, a coming together.
It is a blending of two different genetic stories, you could say, to create a brand new one.
Okay, so who are the key players in this story, these gametes?
Right. So in sexual reproduction, we have the male gametes, which are called sperms.
Gerberms.
And then we have the female gametes, known as ova, or more commonly, we just call them eggs.
Okay, sperms and ova, or eggs.
Yep. And each of these tiny specialized cells carries exactly half the genetic information needed to create a new individual.
Yeah.
They're designed for basically one purpose, to meet and merge.
And that moment, that meeting and merging, that's where the magic really begins, right?
Absolutely. When a sperm and an ovum fuse together,
Yeah.
that specific event is called fertilization.
Fertilization,
Key term.
very key. And when they fuse, they form a single brand new cell. This newly fertilized cell is called a zygote.
Zygote. Okay.
And this microscopic zygote, that's the very, very beginning of a brand new individual. It's like the initial spark of life for every sexually reproducing organism, including us. Wow. So from that one single cell, all the complexity unfolds.
Everything. Every part of that new organism. And that fusion, that combining of the genetic material from both parents, that's profoundly significant.
Why is that?
Well, it's precisely why a new individual inherits characteristics from both parents.
Okay. Makes sense.
It's why you might have, say, your mother's eye color, but your father's hair type. Or why siblings, even though they have the same parents, always look a bit different, right? They have unique combinations of traits.
Right. You see those family resemblances, but everyone's still unique.
Exactly. This blending of genetic information is what drives diversity within a species.
Okay.
It makes each individual distinct. It's like shuffling the genetic deck constantly. It's basically the engine of evolution.
Okay, that covers sexual reproduction. Now let's pivot to the other side of the coin, asexual reproduction.
The single-parent strategy.
Right. In stark contrast, asexual reproduction involves only a single parent. There's absolutely no need for gametes to fuse, no sperms, no eggs meeting.
No male and female involvement in that same way. It's a completely different biological approach.
So how does it work then? What's the advantage?
Well, often this method is much more straightforward and remarkably rapid. It's a quick way for an organism to just make copies of itself.
Copies? Like clones?
Essentially, yes.
Yeah.
Genetically identical copies. Think about the efficiency. If you don't have to spend time or energy finding a mate.
Which can take a lot of effort.
Definitely.
Mm.
Or developing complex gametes and then waiting for them to fuse.
Mm-hmm.
You can reproduce much, much faster.
Okay, so speed is a big plus.
It can be a huge advantage, especially for organisms living in stable environments where conditions are good, or if they need to quickly colonize a new area. It's more about quantity and speed rather than genetic variation.
Right, fascinating contrast. Okay, now that we've got the two big strategies laid out, let's zoom in a bit. Let's really unpack the details, the intricacies of sexual reproduction.
Okay, let's do it.
And we'll use the human body as a familiar example because, well, it illustrates these complex systems really clearly. First up, the male reproductive organs. What are the key parts we need to know?
Okay, so in human males, the main components are the testes. And just a quick note on the word. Testes is plural. The singular is testes. So there are typically two of them.
Right, a pair of testes.
And,
Who else?
And the penis.
Yeah.
So the testes are these really remarkable, highly specialized organs. You can think of them as the factories
Factories for
making those male gametes. We just talked about the sperms. They are produced continuously in the testes. It's like an ongoing biological production line.
continuously producing sperm. Okay. And what about the sperm cell itself, if we could look at one under a microscope?
Yeah, they have a truly remarkable design. It's like a marvel of microscopic engineering, perfectly built for its one job,
Which is?
to swim and find the egg. And despite its unique appearance, a sperm is actually just a single cell, highly specialized, of course.
Okay, a single cell. What are its main parts? I think there are three.
That's right. You got the head, the middle piece, and the tail.
Head, middle piece, tail. What does each part do? Let's start with the head.
Okay, the head. This is where all the crucial genetic information from the male parent is packed, tightly packed inside the nucleus. This is the genetic blueprint that needs to get to the egg. It's like the cargo.
The genetic payload. Got it. What about the middle piece?
The middle piece. Now, our source doesn't name the structures, but this section is absolutely crammed with energy-producing components. Think mitochondria, loads of them.
Why so much energy needed?
Because that sperm has a long, tough journey ahead. It needs significant power to swim through the female reproductive tract to reach the egg. That middle piece is like the engine room, providing the fuel.
Makes sense. And finally, the tail.
The tail. It's not just hanging there, it's a microscopic motor. A powerful whip-like structure, technically called a flagellum.
Flagellum. Okay.
And it propels the sperm forward with incredible force and direction. It allows the sperm to swim actively, vigorously towards the egg.
So without that tail.
No chance. It simply couldn't navigate its way. Fertilization wouldn't happen. That tail is absolutely vital. It's a perfect example of structure matching function in biology.
Really cool. Okay, so that's the male system. Now let's look at the other half of the equation. The female reproductive organs in humans.
Right. So the essential organs here include a pair of ovaries, the oviducts, which you might also hear called the fallopian tubes.
Oviducts or fallopian tubes, same thing.
Yep. Same structure, two names. And then there's the uterus.
Ovaries, oviducts, uterus. Okay, what do the ovaries do? Are they like the testes?
In a way, yes. Just as the testes produce sperm, the ovaries are the primary site for producing the female gametes, the ova, or eggs.
Okay. And is it continuous, like sperm production?
Not quite. In human beings, the process is more cyclical. Typically, a single matured egg is released from one of the ovaries into one of the oviducts approximately every month.
The monthly cycle.
Exactly. It's a rhythmic, precise release. Part of the female reproductive cycle, preparing the body each month for the possibility of receiving a fertilized egg.
And if an egg does get fertilized, where does it go? What about the uterus?
The uterus. If fertilization happens, the uterus becomes absolutely pivotal. It's this incredibly dynamic, muscular, nurturing environment. It's specifically designed to support and protect the developing new life.
So the baby develops inside the uterus.
That's right. From that single fertilized cell, the zygote, it implants in the uterine wall and transforms and grows within the uterus over, about 9 months, drawing all its nourishment and protection from the mother. It's really one of the most amazing processes in biology.
Incredible. Something that often surprises people is the range of egg sizes in the animal kingdom.
Oh yeah, it's huge.
We talk about eggs, and maybe you picture a chicken egg, right?
Probably the most common image.
But human eggs, for instance, they are tiny, very, very small. So small you can barely see them without a microscope, like little specks, but packed with potential.
Microscopic, really.
Yeah.
But then, like you said, you think about the hen's egg. Much, much larger. Easily visible. We eat them.
Right. And then, for a truly mind-boggling scale, the ostrich egg.
Whoa!
The biggest of all. It's the largest single cell known, basically. The largest egg of all can weigh what? Like 3 pounds? Roughly the size of a grapefruit.
Incredible. And this huge range in size isn't just random, is it?
No, absolutely not. It demonstrates the vast diversity in reproductive strategies. A tiny human egg relies on internal development, getting nutrients from the mother,
Right.
while that huge ostrich egg has to contain all the nourishment needed for a chick to develop fully inside it before it hatches. Different strategies for different animals.
Really makes you appreciate the different ways life figures things out.
Totally. Okay, so we've got the gametes produced, the systems are ready. Now we arrive at that crucial moment, the moment of fertilization, the actual beginning of a new individual.
Let's revisit the definition. Fertilization is that fundamental process where a sperm and an ovum come together.
The meeting.
The meeting and the fusion of those two specialized cells we've been talking about. They unite to form a completely new genetic identity.
How does it actually happen? Do lots of sperm get in?
Well, it's often a bit of a race. When numerous sperms, often millions, encounter an egg, it's frantic. But typically, only one single sperm successfully penetrates the egg's outer layers and fuses with it.
Just one out of millions.
Usually, yes. The egg has mechanisms to prevent more than one from getting in, most of the time. And during that critical fusion, the nucleus of that successful sperm merges with the nucleus of the egg.
So the genetic material combines.
Exactly. They become one, forming a single, unified nucleus. And that signifies the start of a completely new genetic blueprint.
And this resulting single cell, the one with the combined genetic material, that's the zygote again, right? Or fertilized egg.
Spot on, zygote or fertilized egg. And this tiny cell, it really is the blueprint. From this single microscopic start, all the complexity of a new organism will arise. Every organ, every tissue, every characteristic, it all stems from this first spark.
And because it's got genetic material from both sides.
Right. That fusion means the new individual inherits characteristics from both the mother and the father.
Which brings us back to family resemblances.
Exactly. It's why you might inherit your mother's laugh or your father's nose or that specific hair color that runs in the family. It's genetics in action, right from that very first cellular union. It creates that incredible diversity within families and within whole species.
Okay, so fertilization is key, but where does it happen? I think there are two main ways, location-wise.
That's right. This distinction is really important because it tells us where this critical fusion actually takes place. First, we have internal fertilization.
Internal, so inside.
Inside the body. Internal fertilization, as the name suggests, occurs inside the body of the female parent.
What's the advantage of that?
Protection, mainly. It offers a highly protected and controlled environment for the egg and sperm to meet. This significantly increases the chances of successful fertilization compared to just releasing gametes out into the open.
Ah, okay. Safer for the gametes.
Much safer. It's a highly evolved strategy, very common in animals that live on land.
Like us.
Like us. Think about humans, cows, dogs, even birds like hens. They all use internal fertilization. It ensures that delicate fusion process happens in a safe place, leading to higher success rates, especially for those early stages.
Okay. And speaking of internal fertilization, this leads to something really interesting, that application of science. Test tube babies.
Right. Or, the more accurate scientific term, in vitro fertilization, IVF.
IVF. Why would someone need this? What problem does it solve?
Well, sometimes for some women, the oviducts, remember those tubes where the egg travels and where fertilization usually happens naturally.
Right, the fallopian tubes.
Right. Sometimes they might be blocked or damaged. And if they're blocked, the sperm simply can't reach the egg.
Ah, so natural conception is impossible.
It can be, yes. And that can be incredibly difficult for couples wanting children.
So what's the solution with IVF?
It's quite amazing, really. Doctors can collect freshly released eggs from the woman's ovaries and also collect sperms from the man. Then these gametes, the eggs and sperm, are carefully brought together in a highly controlled laboratory environment.
Outside the body.
Outside. In a dish. Exactly. That's what in vitro means, literally in glass, referring to the petri dishes or test tubes they used originally. They keep the eggs and sperm together for a few hours under precisely controlled conditions that mimic the body to encourage fertilization to happen right there in the lab.
Wow. And if it works, if fertilization occurs?
If it's successful, the resulting zygote isn't immediately put back. It's allowed to develop for about a week, right there in the lab.
A whole week. What happens during that time?
It starts dividing. One cell becomes 2, then 4, 8. It undergoes those initial rapid cell divisions, forming a very early embryo, just a tiny cluster of cells.
Okay. And what?
After about a week of this initial development, this tiny embryo is then very carefully and precisely placed into the mother's uterus. The uterus has usually been prepared hormonally to receive it.
Ah, so it's transferred to the uterus.
Yes. And from that point on, if everything goes well, the complete development of the baby takes place normally within the uterus, just like any other pregnancy conceived naturally.
So it grows and develops inside the mother.
Absolutely. Grows, develops, and is born just like any other child. That's why the term test tube babies is, well, a bit misleading.
How so?
Because babies don't actually grow in test tubes. Only the very initial fertilization and the first few cell divisions happen in glass. The vast majority of development happens inside the mother's uterus. The test tube is just the starting point.
Gotcha. It's an amazing technology, though, helping where nature faces a block.
Truly profound. A great example of science supporting life.
Okay, so that's internal fertilization and IVF. What's the alternative?
The alternative is external fertilization.
External, so outside the body.
Exactly. This is where the fusion of the male and female gametes takes place outside the body of the female parent.
Where would that happen?
Usually it happens in water, an aquatic environment, which presents a completely different set of challenges and requires different strategies.
Makes sense. Water carries things away. So what kinds of animals do this?
It's very common in aquatic animals. Think about fish, starfish, classic examples. But a really fantastic everyday example, one you might have even seen, is the frog.
Frogs. Right, let's walk through the frog life cycle example. How does external fertilization work for them?
Okay, so picture this. During the spring or rainy season, frogs and codes often migrate, sometimes in large numbers, to ponds or slow-flowing streams.
Heading for the water, their breeding grounds.
Precisely. The water provides the perfect stage for external fertilization. Once they're there, the female frog lays her eggs. And not just a few hundreds, sometimes thousands of them.
Wow, hundreds or thousands. Why so many?
It's a numbers game, really. With external fertilization in an open environment, many eggs won't survive or get fertilized. Laying a huge number increases the odds that at least some will make it.
Okay. And these frog eggs, are they like hen's eggs with shells?
No, not at all. Unlike a hen's egg, a frog's egg is very delicate. It's not covered by a hard shell. Instead, there's a protective layer of jelly surrounding each egg, and this jelly often holds large clusters of eggs together.
What does the jelly do?
It offers some protection, some cushioning, prevents them from drying out too quickly if water levels drop slightly, and keeps them grouped together.
Okay, so the female lays the eggs in the water. What does the male do?
As the female is laying the eggs, the male frog, who is often clinging to her back in a process called amplexus, deposits his sperms directly over the eggs in the water.
Right over the top.
Yep, it's this synchronized release, so a sort of natural ballet happening right there in the pond. The sperms, with their long tails, then swim randomly through the water.
Powered by those little motors we talked about.
Exactly, swimming around, and when they, by chance, come into contact with the eggs, fertilization occurs.
So it's literally fusion happening out there in the pond water.
That's external fertilization in action. It's definitely a game of chance, but by releasing so many eggs and so many sperms, nature makes sure the probability of success is high enough for the species to continue. Amazing, really.
It really is. Okay, so whether it's internal or external, fertilization happens, we get a zygote. What happens next? Let's trace that incredible journey from zygote to fetus.
Right. So following that crucial moment of fertilization, the zygote starts an amazing process of repeated cell division. It just divides and divides and divides.
One cell becomes 2, 2 becomes 4.
4 become 8, 16, 32. It multiplies exponentially, rapidly. and in a very organized way.
And this ball of dividing cells is called?
This process forms an embryo. That's the term for the developing organism in its very early stages. It starts as a simple bundle of cells, but very quickly those cells start to differentiate. They start to become specialized, forming the basic tissues and structures of the organism.
Like a blueprint starting to take shape.
Exactly. Like a tiny living sculpture being formed from within.
Yeah.
Now, what happens next depends on the type of fertilization and the animal.
Okay, so for internal fertilization, like in humans.
Right. In animals with internal fertilization, the embryo usually undergoes a critical step called implantation. It embeds itself into the wall of the uterus.
Attaching to the mother.
Yes, establishing that vital connection. This is where it continues its development, drawing all the nutrients, oxygen, and protection it needs directly from the mother's body. It allows for a long, sustained, protected growth, period.
Okay, what about animals like hens? They have internal fertilization, but lay eggs.
Good point. For animals like the hen, the development process starts internally, but finishes externally. After internal fertilization, many protective layers, including that hard shell, are formed around the developing embryo while it's still inside the mother's oviduct.
Ah, the shell forms inside her.
Yes. Then she lays the egg. But the job isn't done. The hen then typically provides consistent warmth by sitting on the egg, incubating it for about 3 weeks.
Why is the warmth so important?
That warmth is absolutely crucial. It maintains the optimal temperature needed for the chick embryo to continue developing fully inside the eggshell. It keeps all the chemical reactions of life going at the right pace.
And after 3 weeks?
Once it's completely developed, the chick uses a small egg tooth on its beak to peck and break open the shell and emerges, having done all that incredible transformation inside the egg.
Amazing. Now, what about external fertilization, like our frogs? Where does the embryo develop?
In animals with external fertilization, like frogs, the embryo develops entirely outside the female body. It develops within those jelly-like egg coverings, submerged in the water.
So it develops right there in the pond?
Yep. Once the embryo inside the egg is sufficiently developed, the eggs hatch. And what emerges isn't usually a miniature adult.
Right. For frogs, it's the tadpoles.
Exactly. Young forms like tadpoles are released. And these often look drastically different from the adults, which hints at that transformation process we need to talk about.
Okay, one more term first. As the embryo keeps developing, whether inside the mother, inside an egg, or in the water, there's a point where it becomes more recognizable, right?
Yes. The stage of the embryo where all the major body parts are identifiable. You can clearly see a head, limbs starting to form, the basic organ structures are laid down. That stage is called a fetus.
Fetus. So embryo is the very early stage. Fetus is when things look more formed.
That's a good way to think about it. Fetus implies a more advanced stage of development where the basic body plan is clearly established. That tiny zygote you started as eventually grew through the embryonic stage and then the fetal stage to become, well, you.
Incredible journey. This actually leads us nicely into thinking about the two major ways animals bring their young into the world: viviparous versus oviparous animals.
Right. It boils down to live birth or egg laying.
Let's start with viviparous. What does that mean?
Viviparous animals are those that directly give birth to live young ones.
Live birth, like mammals.
Mostly, yes. Their offspring develop fully, or almost fully, internally within the mother's body, usually in the uterus, drawing that continuous nourishment and protection. They are born alive.
And are they usually quite developed when they're born?
Often, yes. More developed than hatchlings from eggs. They might be capable of some independent activity quite quickly, although they usually still need significant parental care. Think about familiar examples. Human beings, cows, dogs, cats, whales.
All give birth to live young. Viviparous.
Their young, developed internally, complete that fetal stage inside, and are born live.
So what's the opposite? Oviparous.
Exactly. Oviparous animals are those that lay eggs.
Egg layers.
Yep. The young ones then develop inside those eggs, which are usually laid outside the mother's body, and eventually they hatch from the eggs.
So most birds, reptiles, amphibians.
Absolutely.
Yeah.
Hens, frogs, lizards, snakes, turtles, fish, insects like butterflies. Classic examples of oviparous animals. You've probably seen frog eggs in a pond, or maybe found a bird's nest with eggs, or even a lizard's egg tucked away somewhere.
So the egg provides the protective environment and nutrients for development outside the mother.
That's the strategy. The parent might provide warmth, like the hen incubating, or protection by guarding the nest, or in some cases just lay the eggs and leave them to develop and hatch independently.
So The key difference is where that main period of development happens inside the mother or inside an external egg.
Precisely. Is development internal with continuous support, viviparous, or external within a self-contained egg, oviparous? It's a major strategic split in the animal kingdom, with big implications for parental care and the vulnerability of the young.
Fascinating stuff. Okay, let's move on now to section 3. This is one of my favorites. From young one to adult. The magic of metamorphosis.
Yes, transformation. This is truly captivating. It often sparks so much wonder.
The core ideal here is that sometimes the young forms of animals look so different from the adults. It's almost unbelievable. It's not just about getting bigger.
No, it's a complete overhaul, a radical transformation.
And this incredible biological process where a larva that's the immature, often quite distinct form, transforms into an adult through drastic fundamental changes in its body structure. What's that called again?
That process is called metamorphosis.
Metamorphosis, great word, captures that profound change.
It really does. It's like a biological reinvention of the organism partway through its life.
And the classic example, the one everyone usually thinks of, is the frog life cycle, right?
Absolutely perfect example. If you picture a frog's journey, it shows these incredibly distinct stages so clearly. It starts as an egg.
Laid in the water, as we discussed.
Right, which then hatches into a tadpole. That's the larval stage.
The little swimming thing with a tail.
Exactly. Perfectly suited for life entirely in water. And then, through a series of really dramatic changes, it becomes the adult frog.
Just try and picture the difference. That tadpole. It's small, it lives only in water, it breathes using gills like a fish, it swims using its tail.
Completely aquatic. Eats algae mostly.
And then compare that to the adult frog. It lives mostly on land, breathes air using lungs, has no tail, but has powerful legs for hopping, and eats insects.
It's astonishing. Can you really imagine that same tadpole somehow morphs into that hopping, croaking, land-dwelling frog?
Yeah.
It loses its gills, develops lungs, the tail shrinks away, legs grow. It's like something out of a fantasy story, but it's real biology.
Happens in ponds all over the world. Mind-blowing.
Yeah.
And it's not just visually dramatic, right? There are ecological reasons for this.
Oh, absolutely. There are significant ecological implications. Metamorphosis allows the different life stages, the larva and the adult, to occupy completely different ecological niches.
Meaning they live in different ways, eat different things.
Exactly. Think about it. The aquatic tadpole eating algae doesn't compete for food with the terrestrial insect-eating adult frog.
So the kids aren't competing with the parents for food.
Precisely. This drastically reduces competition for resources between the young and the adults of the same species. It maximizes the survival chances for the whole population because they're not all trying to eat the same thing in the same place.
That's really clever, a brilliant evolutionary adaptation.
It is. It allows a single species to essentially exploit multiple environments and food sources during its lifetime. Many insects, of course, famously do this too. Think caterpillar to butterfly. Same principle, different players.
Very cool. Okay, let's shift gears now. Back to the other main reproductive strategy. Section 4. Asexual reproduction, the single-parent strategy in action.
Right. We defined it earlier, but let's revisit. Asexual reproduction involves only one single parent.
No fusion of gametes.
Correct. No sperm, no egg, no zygote formed from two parents merging their genes. It's simply one organism making a genetically identical copy of itself.
A clone, essentially.
Yep. And because it doesn't involve finding a mate or the complexities of the gamete fusion, it can be incredibly fast.
So great for rapid population growth.
Exactly. Yeah. If conditions are good, plenty of food, right temperature and organism can quickly produce many, many identical offspring. Huge advantage for colonizing new areas or just multiplying quickly when times are good.
Okay, so what are some examples? How does this actually happen?
Well, a fantastic example is budding in hydra.
Hydra. What's hydra again?
Hydra is this small, really fascinating freshwater organism. You find it in ponds and streams. It's tiny, has a tube-like body with tentacles at one end, and it usually attaches itself to underwater plants or rocks.
Oh, okay. And it reproduces by budding. What does that mean?
It means that new individuals literally develop as outgrowths, like little buds right on the side of the parent hydra's body.
A bud just grows off the parent.
Yeah. Imagine a small bump appearing on the side of the hydra.
Oh.
That bump slowly grows, elongates, develops its own miniature set of tentacles and a mouth.
A mini hydra growing on the side.
Pretty much. Remember how we learned about budding in yeast earlier in your science classes? Where a new yeast cell just pinches off from the parent? It's a very similar principle here. This bud grows, becomes a fully functional miniature hydra, and then eventually it just detaches from the parent.
And swims off on its own.
Well, floats off or moves off, yes. It becomes a completely independent organism. But genetically, it's identical to the parent it budded from. It's like natural cloning on a small scale. Very neat.
Okay, budding in Hydra. Cool. What's another example?
Another really common one, especially in the microscopic world, is binary fission in amoeba.
Amoeba, the little shape-shifting blob we see under the microscope.
That's the one. That fascinating single-celled organism, constantly changing its shape as it moves and eats. Just one single cell.
And it reproduces by binary fission. What does that mean? Fission sounds like splitting.
It is. Binary means two, and fission means splitting. So
Yeah.
binary fission is literally splitting in two. It's a beautifully simple, yet incredibly effective way for single-celled organisms to reproduce.
How does it work? Does it just pinch in the middle?
Essentially, yes. But there's a crucial first step. The process begins inside the parent amoeba, with its nucleus dividing into two. The genetic material duplicates, and the nucleus splits to form two identical nuclei.
Okay, gotta copy the instructions first. Makes sense.
Exactly. Got to make sure each new daughter cell gets a complete set of genetic instructions. Then, this nuclear division is quickly followed by the division of the amoeba's entire body, the cytoplasm, the organelles, everything into two separate parts.
So the whole cell splits.
The whole cell splits. It elongates, constricts in the middle, and eventually pinches off into two distinct halves. Each of these new halves receives one of the newly formed nuclei.
And then you have.
And you have two new independent amoebae, produced from just one parent amoeba. Each one is a complete functional organism ready to grow and potentially divide again.
Wow, that sounds incredibly efficient.
It is. Binary fission is super efficient for single-celled organisms. It allows for really rapid reproduction, creating identical copies very quickly. That's how populations of bacteria and amoebas can explode under the right conditions. It's a fundamental strategy for microbial life.
Simple splitting, okay. Budding and binary fission. Two cool examples of asexual reproduction. Now, this idea of making identical copies, it leads us naturally into our final section, Section 5, the fascinating and complex world of cloning.
Cloning. Taking that idea of identical copies to a whole new level.
When we talk about cloning in this context, what exactly do we mean?
We're talking about the production of an exact genetic copy. It could be a copy of a single cell or another living part like a tissue, or even a whole, complete organism.
Right.
Creating a genetically identical twin, essentially, but often delayed in time.
Right. Not just simple splitting like amoeba, but a more deliberate, often scientifically assisted process to create an exact replica.
Precisely. And it's a truly remarkable scientific achievement, but also, as you said, a complex one. It sparked immense discussion, wonder, and some serious ethical debates, too.
It really pushes the boundaries of our understanding of life and reproduction, doesn't it?
It absolutely does. And the landmark story here, the one that really brought cloning into the public eye, is the story of Dolly the sheep.
Dolly! I remember hearing about Dolly. Who is behind that?
It was the incredible work of Sir Ian Wilmot and his colleagues at the Roslin Institute, which is near Edinburgh in Scotland. Their meticulous research and experimentation led to a truly historic moment.
When was Dolly actually born?
Dolly was born on July 5th, 1996. And she was incredibly significant because she was the first mammal ever successfully cloned from an adult somatic cell.
Okay, wait. From an adult cell. Why is that so important? Hadn't cloning happened before?
Well, yes, scientists had cloned animals from embryonic cells before. But Dolly was different. She was cloned from a cell taken from an adult sheep specifically, a cell from the mammary gland. This was a massive breakthrough because it showed that a specialized adult cell, which had already differentiated to do its specific job, could somehow be reprogrammed, its genetic clock reset, to generate a whole new organism.
Wow, okay. So it proved adult cells weren't locked in forever. How did they actually do it? The process sounds complicated.
It is quite intricate, and it shows the precision needed. Okay, step by step. First, they took that somatic cell, the mammary gland cell, from an adult female Finn Dorset sheep. This sheep was the genetic donor. Her cell contained the complete genetic blueprint for Dolly.
Okay, got the blueprint cell from sheep A, Finn Dorset. What next?
Simultaneously, they obtained an unfertilized egg cell from a different sheep, a Scottish blackface ewe, sheep B.
An egg cell from sheep B. Okay.
Now, here's the crucial bit. They carefully removed the nucleus from this egg cell. Remember, the nucleus contains the egg's own genetic material. By removing it, they essentially created an empty egg cell, a blank canvas ready to receive new DNA.
So they took the nucleus out of sheep B's egg.
Exactly. and a nucleated egg. Then they took the nucleus from the fin-dorsate sheep's mammary gland cell, sheep A's blueprint, and carefully inserted that nucleus into the empty egg cell from the Scottish blackface ewe, sheep B.
So sheep A's nucleus went into sheep B's empty egg.
Precisely. This created a reconstructed egg. It had the cytoplasm and machinery of sheep B's egg, but the complete genetic instructions from sheep A. This was the key genetic transfer step.
Okay, that's clever. Then what? Does it just start growing?
Almost. They needed to give it a kickstart. They gave this reconstructed egg a small electric pulse. This pulse acted like a stimulus, essentially tricking the egg into thinking it had been fertilized naturally. It prompted the egg to start dividing, just like a normal zygote would.
So the electricity made it start developing.
It initiated the first cell divisions right there in the lab dish. This early embryo was allowed to develop for a few days.
And then, where did it go?
Then this developing embryo was implanted into the uterus of another sheep, the Scottish blackface ewe. Sheep B, the original egg donor, or sometimes a different surrogate, this ewe acted as the surrogate mother.
So sheep B carried the pregnancy, even though the genetics were from sheep A.
Exactly. The Scottish blackface ewe carried the pregnancy to term, providing the uterine environment for the embryo, which contained sheep A's DNA, to grow and develop. The embryo developed normally inside her womb, and finally, after a normal gestation period, Dolly was born.
Wow. So Dolly is born from the Scottish blackface ewe. But who did she look like? Who was she genetically identical to?
That's the amazing part. Despite being carried and born by the Scottish blackface ewe, Dolly was found to be absolutely genetically identical to the fin-dorsate sheep, sheep A, the sheep from which the original mammary gland nucleus was taken.
Because all the DNA came from that nucleus.
Precisely. All her genetic information, every trait, came solely from that fin-dorsate nucleus. The Scottish blackface ewe's nuclear DNA had been removed, so Dolly showed none of her characteristics. She was a perfect genetic replica of the fin-dorsate sheep, a true clone.
Incredible. And was Dolly healthy? Did she live a normal life?
Dolly was reported to be a healthy clone initially. And interestingly, she went on to have several lambs of her own, conceived naturally through normal sexual reproduction. This proved that cloned animals could themselves be fertile.
So clones can reproduce normally?
Dolly did, yes. Sadly, though, Dolly died relatively young on February 14, 2003. She was only 6 years old. She had a progressive lung disease.
Oh. Was that because she was a clone?
That's been debated a lot. The lung disease she had is common in sheep, especially those housed indoors. But her premature aging and death raised questions about the long-term health and potential subtle abnormalities in cloned animals. It wasn't definitively linked to the cloning process itself, but it added to the complexities.
Right. So Dolly was this huge success story, but it wasn't all straightforward. Cloning is still difficult.
Oh, absolutely. It's vital to acknowledge the challenges and implications of cloning. While Dolly was a phenomenal success, many subsequent attempts to clone other mammals and even sheep have faced significant difficulties. It is not a simple or highly efficient process.
What kinds of difficulties?
Well, the success rate for creating live, healthy clones from adult cells remains remarkably low across many species. Many cloned embryos fail to develop properly or implant in the surrogate mother.
And those that are born?
Tragically, many cloned animals that are born either die very soon after birth or they're born with severe abnormalities, problems with their organs, their immune systems, oversized bodies. It seems the reprogramming of that adult cell nucleus isn't always perfect.
So lots of things can go wrong.
Lots of things. There are still many unknowns about the precise biological mechanisms involved, why failures happen, and the long-term health and sort of normality of cloned organisms. It involves complex genetics, but also epigenetics, how genes are switched on and off, which seems hard to reset perfectly. We're still learning.
It really does push the boundaries, doesn't it? Cloning. It opens up so many huge questions that go way beyond the science lab.
Totally. What are the ethics of creating exact genetic copies? Should we be doing this? What might it mean for biodiversity? Could it be used to help save endangered species, bringing them back from the brink?
Or could it reduce genetic diversity if we start cloning prize farm animals too much?
Exactly. And what about human cloning? That opens a whole other, even more complex ethical minefield.
Yeah, big questions.
It's a field that's constantly evolving. Research continues, techniques get refined, our understanding deepens. And for you guys, as grade 8 science enthusiasts, you're really just seeing the beginning of understanding its immense potential, maybe for medicine, like cloning tissues for transplants or for agriculture.
But also its significant challenges.
And its significant challenges, both scientific and societal. It's definitely a field to keep an eye on because its implications for the future of life and our relationship with it are potentially vast.
Wow. Okay. What an incredible deep dive that was. We've covered so much ground today exploring the astonishing world of animal reproduction.
We really have. We started with that fundamental drive, the need for species to continue. Then we went through the intricate dance of sexual reproduction,
Gametes, fertilization, zygotes,
the amazing transformations of metamorphosis,
tadpoles to frogs,
then the surprising simplicity and speed of asexual reproduction.
budding and binary fission.
And finally, we touched on the truly mind-bending reality and the profound questions raised by cloning.
It really covers the whole spectrum, doesn't it? Remember, whether it's that microscopic fusion of tiny gametes happening inside an animal or out in a pond,
Or the incredible development of an embryo inside a mother or an egg.
or that complete transformation of a larva into an adult. Reproduction is how life finds a way. It adapts, it innovates, it ensures continuity.
It's this fundamental process that is constantly renewing and shaping the incredible biodiversity we see all around us on our planet. It's happening right now, all the time, in every ecosystem. Life ensuring life continues.
And as your grade 8 science teachers here at School on X, we really, really hope this deep dive has sparked your curiosity. We hope it's given you a much clearer, more detailed understanding of how this essential process works.
Because it impacts every living thing around us, including us. Understanding reproduction is key to understanding life itself.
Definitely. So let's leave you with a final thought to mull over, a provocative question, perhaps.
Okay, get us with it.
If reproduction is so absolutely vital for species to continue, and given this incredible variety of methods we've explored today, from simple splitting to complex cloning, what might the future hold? Especially now, as global environments are changing so rapidly.
With challenges like climate change, habitat loss, how might reproduction adapt?
Exactly. Will we see species naturally evolving new, perhaps even more unusual reproductive strategies to cope with these new pressures? Faster reproduction, different timing,
Yep.
stranger methods.
Or will there be an increasing reliance on human intervention? Things like IVF for endangered zoo animals, or even cloning maybe, as a last resort for conservation to try and save species on the very brink of extinction.
And if we do rely more on things like cloning for conservation, what impact might that have on the natural world as we know it? Does it fundamentally change what wild means? Does it solve the underlying problems? Does it affect genetic diversity in the long run?
Big questions. Where do we draw the line between helping nature and controlling it? What are the unforeseen consequences?
Lots to think about there. Keep those questions buzzing in your mind. It shows you're thinking like a scientist.
Absolutely.
Thanks so much for diving deep with us today.
Until next time.
Keep exploring, keep questioning, and keep learning.